So now let's look at the leukocytes, or the white blood cells. They're the most diverse, but the least abundant formed element. Remember, there's only about 5,000 to 10,000 white blood cells per microliter of blood. The white blood cells are primarily involved in protecting against infectious microorganisms and other pathogens. They have very conspicuous nuclei. They spend only hours in the bloodstream before they'll migrate to the connective tissue. They retain all of their organelles in order to perform protein synthesis. All white blood cells have granules that are formed from lysosomes called nonspecific granules. They're inconspicuous, so the cytoplasm does look clear. Granulocytes have specific granules that contain enzymes that are employed in defense against pathogens or invading particles. The granulocytes are broken down into three types. There's neutrophils, eosinophils, and basophils. The majority of granulocytes are neutrophils. They're polymorphonuclear leukocytes. They're barely visible granules in the cytoplasm, and they have a 3 to 5 lobed nucleus. You can see the neutrophils here in this first figure. Eosinophils, on the other hand, are only about 2 to 4%. They're large, rosy orange granules, and they have a bilobed nucleus, so you can see the granules more clearly. The basophils here on the right are large and abundant violet granules. They have an obscure S-shaped nucleus you can sort of make out in here. The agranulocytes, still having granules but not ones that we can really see, are called lymphocytes and monocytes, the majority being lymphocytes that we see here on the left. They have various amounts of bluish cytoplasm and some ovoid, round, uniform dark violet nucleus. The monocytes are the largest white blood cell. They have an ovoid, kidney, or horseshoe-shaped nucleus. Now, the functions of each of these are very interesting. The neutrophils increase in numbers during bacterial infections. They're involved in phagocytosis of bacteria, and they'll release some antimicrobial chemicals. The eosinophils increase in number in parasitic infections, so collagen diseases, allergies, and diseases of the spleen and central nervous system as well. They are involved in phagocytosis of antigen-antibody complexes, also allergens, and some inflammatory chemicals. They release enzymes that destroy large parasites. The basophils increase in numbers in situations like chickenpox or sinusitis and diabetes. They secrete histamine, which is a vasodilator. It speeds blood flow and helps repair the injured area. They also secrete heparin, which is an anticoagulant, and this promotes mobility of other white blood cells in the area because the blood is thinner. The agranulocytes, the lymphocytes and monocytes, are also involved in immune response and inflammation. The lymphocytes increase number in infections and immune responses. They destroy cells like cancer cells and foreign cells and virally infected cells. So lymphocytes are very important in the prevention of cancer. Believe it or not, our own bodies can fight cancer. The lymphocytes will present their antigens and activate other immune cells. They basically say, here's a cancer cell, here's a virally infected cell, and they label it so that the cleanup crew can come along later and sweep them up. They coordinate the action of these other immune cells, and they also secrete antibodies that provide immune memory. We'll investigate all of these functions when we look at the chapter on the lymphatic system and immunity. Monocytes are the other agranulocyte. They are increased in numbers in viral infections and inflammation. They're going to leave the bloodstream and transform into macrophages in the tissue. There, they'll phagocytose the pathogens and debris. They present antigens also to activate other immune cells. They are antigen-presenting cells. So take a moment here and pull out a piece of paper and write down all the different types of white blood cells and what their functions are. Briefly, as much as you can remember. And then go into your notes, fill in the gaps and details, and work that brain on a little bit of recall. Now we'll investigate the leukocyte life cycle. The production of white blood cells is called leukopoiesis. There are pluripotent stem cells, PPSCs, here, and they will form myeloblasts that form neutrophils, eosinophils, or basophils, depending on what receptors they have on their cell surface. They will also form monoblasts that will form the monocytes, and lymphoblasts that give rise to all forms of lymphocytes. These committed cells have receptors for colony-stimulating factors. The mature lymphocytes and macrophages will secrete several types of colony-stimulating factors in response to various infections and immune challenges. Each colony-stimulating factor stimulates a different white blood cell to develop in response to its specific needs. Thus, a bacterial infection can trigger the production of neutrophils, whereas allergies trigger the production of eosinophils. The red bone marrow stores the granulocytes and the monocytes until they're needed and contains about 10 to 20 times more of these cells than the circulating blood does. All lymphocytes begin developing in the bone marrow, but they don't stay there. Some types mature there and others migrate to the thymus, like the T lymphocytes, to complete their development. The granulocytes will leave the red bone marrow in about 8 hours and live 5 days longer. The monocytes will leave in about 20 hours, and there they'll transform into macrophages, and they'll live for several years. The lymphocytes will provide long-term immunity, decades, so they live a very long time, being continuously recycled from blood to the tissue fluid to lymph and back to the blood. As with red blood cells, there are many white blood cell disorders. Leukopenia is when we see a low white blood cell count, one that's below 5,000 per microliter. The causes of this low white blood cell count are often radiation or poisons or infectious disease. The effects are to elevate the risk of infection. This is why radiation therapy actually acts to suppress the immune system. And to me, it's surprising that it's used because our immune system itself, when it's healthy, can fight cancer. If we kill the leukocytes, then how can they possibly help us fight cancer? Another interesting topic you might bring to the end of the semester presentations. Leukocytosis is when we have too high of a white blood cell count, say above 10,000 per microliter. The causes of this are infection, allergy, and disease. The differential white blood cell count can identify what percentage of total white blood cell count exists of each type of leukocyte. This can give us some insight into what's going on when someone is sick. Leukemia is cancer of the hemopoietic tissue. This usually produces an extraordinary high number of circulating leukocytes and their precursors. That is, the hemopoietic tissue is producing more white blood cells than it should be. Myeloid leukemia is when we see uncontrolled granulocyte production. Lymphoid leukemia is when we see uncontrolled lymphocyte or monocyte production. Acute leukemia appears very suddenly and progresses rapidly, and death is often imminent within months. Chronic leukemia may be undetected for months. The survival time can be up to 3 years or more. The effects when normal cell percentages are disrupted, we'll see impaired clotting function and the onset of opportunistic infections because the white blood cells are just not prepared to fight. These are slides of normal and leukemic blood. The top one is a normal blood smear, and the bottom one is blood from a patient with acute monocytic leukemia. Note the abnormally high number of white blood cells, and especially the monocytes in this figure. So that brings us to the end of the section on leukocytes. Be sure that you have them straight before we move on to platelets and homeostasis and the control of bleeding.